our next group of speakers is going to be Hannah King and Maddison Hicks from the University of Washington, as well as Emilio Tripp from the Karuk Tribe, talking about one of our favorite topics at KBMP, beavers. Hannah, Maddison, and Emilio, take it away, please.
Hi, thank you. We actually had a switch, so Emilio will not be joining us today, but we do have Jessica Conrad who will be joining us. So I will pull up my screen shared now. Oh, actually.
You're muted, Maddison. The fun of remote presentations. Maddison, Hannah.
All right, we're getting that pulled up now. Thanks for your patience, everybody.
Yeah. All right. Progress. And we see your full screen. Looks great.
Okay, Jessica, would you like to start?
Yeah, sure. Ayuki Kora, Nanithuti Jessica Conrad, Na Karuk Arara. Hi, everyone. Hope you all are well. My name is Jessica Conrad. I am of the Karuk people, upriver people, and I work for the Karuk Department of Natural Resources Wildlife Program. And I'm always feel extremely fortunate to be able to do this work, and glad I get to introduce this project to you all. The Karuk people have fought to and continue to fight to live in this in our ancestral territory and to have our connection to the natural environment in this place. And it's vital to our culture and our sovereignty that we're leading efforts like the Sahpihnîich project on the Klamath. And the word Sahpihnîich is a word for beaver in Karuk, and it translates to the old man of the river, which is fitting as they're known to rely more on their wits and their hard work for their survival than their brute strength and fierceness. Sahpihnîich is a culturally significant species for the Karuk people, and mainly due to their role that they play in the ecosystem of the watershed and their ability to provide habitat for juvenile salmon, which are integral to our culture and one of our main food sources. And there really hasn't been a lot of data collected on them through the years. So this project has been basically a pilot project for implementing different methods of gathering that data. And it's been in partnership with these awesome folks from the University of Washington. And it's really, having that partnership with them has really taken this project to a new level and elevated the effort and resources that we've been able to put into learning about beaver on the Klamath. And yeah, I'm just excited for them to share with y'all about this project.
Thanks, Jess. So as Jess mentioned, this project was done in partnership between the Karuk Tribe Department of Natural Resources Wildlife Division and the Fresh Lab at the University of Washington, which is where Hannah and I are getting our master's degrees at the School of Marine and Environmental Affairs. So really at the foundation of this project was our collaboration with the Karuk Tribe. We really wanted to ensure that we were co-producing knowledge in a way that honored data sovereignty and respect for the tribal collaborators. And this involved going through a process called practicing Pikyav, which essentially serves as a tribal institutional review board. And throughout the process, we outlined in detail exactly how data was going to be collected, used, protected, and made accessible to our collaborators throughout the entirety of the project. And it really goes into showing how important it is to have Karuk people doing Karuk work and to incorporate Karuk language into projects like this. So this June, Hannah and I were fortunate enough to make a trip to Karuk Aboriginal Territory, which is indicated on the map on the right, to assist with data collection for this project. The map on the left shows the entire Klamath River watershed, and it also indicates where there are 2 key hydroelectric dams, which is something we're going to come back to later. So as Jess mentioned, this project was focused on piloting research methods to help the Karuk tribe better understand the current distribution of beavers in the mainstem and select tributaries. And we were really focusing on beavers specifically because they once thrived in the area, but after the fur rush and the gold rush that happened when settlers arrived to the area, their populations were really decimated. And beavers are culturally significant species and are keystone species and ecosystem engineers that show great potential for restoring streams and riparian ecosystems, especially when it comes to providing habitat for critically the ESA listed coho salmon. So there were a lot of different research questions that we were hoping to answer throughout this process, but we were primarily interested in discovering the best methods for collecting and analyzing data about beaver populations. We also wanted to understand where community members have been seeing beavers, but piloting those methods in the field was definitely our top priority. We really wanted to see what the best data collection methods were and to understand how to focus our future efforts. So this is an evolving process, and throughout this, we are really open to feedback and any recommendations because this project is going to continue beyond what we're presenting today. So we used a lot of different methods in this project that kind of all came together really well to complement each other and give us a really broad scope of what efforts need to be taken for beaver research. So first, we did a habitat suitability model using the Beaver Restoration Assessment Tool, or the BRAT model, to predict where we were most likely to see signs of beavers. And we let this model inform our data collection in the field alongside traditional ecological knowledge and the local knowledge of our collaborators. So in the field, we did environmental DNA collection as well as river floats and walking surveys, and also did a couple of informal interviews and surveys with Karuk tribal members, and the wildlife crew was also able to deploy a couple of game cameras. So as I mentioned, we used the BRAT model to help inform where we collected data, and that's kind of where these images came from. So the image on the right is a map that shows the amount of frequent and pervasive existing dam habitat. The blue and green indicate stream reaches where the model predicted an existing dam capacity of 5 dams per kilometer or more. And the graph on the left is a calculation of the mileage of that dam capacity within the watershed. And the highlighted creeks are the ones that we ended up doing our sampling on. So eDNA sampling was done using kits from a company called Jonah Ventures to look for recent beaver DNA that's been left behind in the water column. And it serves as a really short-term recent snapshot of the presence and absence of beavers in the area. So basically we took a giant syringe full of water and just shot it through a filter that then was sent back to the lab to test for that beaver DNA. So here you can see Hannah in the field doing some of that sampling. We also conducted river floats and walking surveys, and during our river floats, we got in inflatable kayaks and surveyed select sections of the Klamath mainstem. We ended up doing a total of 3 river floats and 1 walking survey on Red Cap Creek, and we did a walking survey there because it was an area that had previously known to have a beaver den. So some of the signs that we were looking for during these surveys included beaver slides, bank dens and lodges, broken trees, fecal matter, trails, dams, scent mounds, and gnawed sticks and branches. And of course, we were looking for beavers as well, but unfortunately, we did not see any this summer. When we did see signs of beaver activity, we made sure to document it using a Garmin GPS system and took really detailed notes about everything that we were seeing. So this is an example of some beaver chew on some willows off the Klamath mainstem that we saw during our river float. And you can tell that this is definitely beaver because they are really clean cut gnawed branches. But these look like they might be a little bit older. Whereas if you look at these photos, the wood is a little bit darker, which kind of indicates that it might be fresher beaver chew. And that's really what we were mainly looking for because we wanted to get an idea of the very current and recent idea of where beavers are located. And I think at the site where these photos were taken is actually a spot where they were able to deploy some game cameras as well.
All right, so now to take a look at the results of these different data collection methods. So this slide just shows, or this map shows a visual of areas that we did survey. So the circles show the eDNA sampling sites throughout the area, and the blue lines show our walking and float surveys. And then Karuk Aboriginal Territory is marked as the black boundary there. Okay, so to take a closer look at the results of our float and walking surveys, we ended up surveying in total about 17.5 miles of river. And within that area, we identified 16 active beaver sites, including one bank den along the main stem of the Klamath, which was an exciting find. And this map just shows a little more detail of our float surveys. So you can see the first 3 float surveys in sort of the mid-Klamath area were the ones that were conducted with the Karuk Wildlife Division. And then 2 additional float surveys were conducted in the upper Klamath, tagged along and helped Laurel with her algal surveys. We also surveyed for beaver there. And so along this map here, you can see the red dots are marking active beaver signs that were identified along the way. And this just shows a little bit more detail about each of these reaches. So this shows each survey reach, the distance surveyed in miles, the number of beaver sign observed within that reach, the sign quality, number of surveyors, and estimated time. And just a couple of things to highlight here. The variability in the amount of beaver sign found was actually quite high. So for example, here in Sluice Box to Portuguese Creek Reach, which was about a 3-mile river section, we only identified 1 active beaver site versus this Brown Bear to Horse Creek section, which was a slightly shorter reach and had 4 very active distinct beaver sites. Another thing to note here is just the amount of time that it did take for these surveys. So this was just an estimation, but we estimate about 24 hours for about the 17.5 miles surveyed. So looking at our eDNA results, in total, we sampled 18 separate sample sites and 50 samples were collected overall. So we did 3 replicates per sample site and then had sort of 2 additional tests that we did just out of curiosity for some sites that we had some interest in. And so the results of all this sampling is shown on this map here. You can see the light yellow circles show areas or sample sites that came back with 0% detection for beaver DNA. This orange color shows sites that came back with partial detection for beaver DNA, and the red circles show 100% detection of beaver DNA for all 3 samples at each site. So the 2 sites that we got 100% detection at were our control, which was the Sugar Creek Habitat Restoration Site in Scott River Valley, where there was a known active beaver lodge, and we took a sample directly from the pond in front of that. And the other site with 100% detection was the mouth of Beaver Creek. These 2 sites with partial detection rates were interesting to us. This site here at Rogers Creek, this result can be explained by some known error that occurred during sampling. This sample was noticed to have actually been taken from an area where Rogers Creek was already mixing with the main stem of the Klamath, and that was the sample that came back with this partial positive. This same result in Horse Creek was found, and this one we don't really see any mechanism for cross-contamination. So this is an interesting area that we'd like to look into further and potentially do some surveys along that area to see if we do indeed see beaver sign. So just a quick comparison here of these different methods and some recommendations for moving forward. The float and walking surveys are very resource intensive and time consuming, and yet we did determine that they were the most thorough method for surveying, and they give us the most information about the ecosystem as a whole. eDNA, we believe, can be very helpful as sort of a broad brush tool and used to identify sites for further surveying, because you can quickly cover much more ground with eDNA. It only takes about 30 minutes per site to do the water sampling, and it can be used for areas that would be very difficult to access via float or walking survey. However, in order to really accurately interpret the results of our eDNA testing, there are many remaining questions, such as how long does beaver DNA persist in the water? And based off of the literature, we know that this is variable and dependent on many different parameters, such as pH and temperature and stream flow. And based off of those parameters, the answer can change from hours to weeks. Similarly, the range of detection is also quite variable, from about 100 meters to 3 miles, also dependent on stream parameters. And so really gaining a better understanding of the eDNA and will allow us to better interpret those results moving forward and to better use that tool. Another thing to note is that eDNA only detects recent activity, which is what we were looking for. However, our walking survey on Red Cap Creek, we did find an abandoned beaver dam that would not have been shown by the eDNA and still provided a lot of useful information about that habitat. Community sightings moving forward, we think this could be a really powerful tool as well to aid in our population assessment. From the interviews and conversations that we did have, there was a lot of knowledge within the community, but there's a need for a more streamlined method of data collection for this. And some things we're looking into for the future are more formalized surveys, or possibly the adoption of the use of a tool, an existing tool like the app iBeaver that allows folks to crowdsource basically beaver sightings and upload photos and the timing and location of sightings. So next steps, the KDNR Wildlife Division has secured additional funding for spring and summer walk and float surveys. And so at this point, we're really trying to decide upon what the best methods and best practices for those surveys are, and also identifying major gaps in our current data collection and using this pilot project to really inform where we study next. And additionally, really honing in on what additional data is needed to conduct a population estimate. And in that light, we're trying to move from this sort of presence absence data collection that we used during this survey to gain more information on the actual density and distribution of the beaver population along the Klamath. To this end, we're also looking into some other methods, potentially hair snares or mark and recapture programs that could help inform that population estimate.
So overall, this work is really important for dam removal because it allows us to standardize the float and walk surveys that we can use to monitor populations after dams are removed. It's also really important to have data both pre and post dam removal because that opens avenues to gather evidence to support future dam removal efforts. And kind of like we mentioned, this is a very evolving process. We're really kind of open to any sort of feedback that folks might have or recommendations to move forward. And these are our various contact information, and please feel free to reach out to any one of us about this. And we're hoping to continue this project in the future. So thank you, everybody. And we are open to questions now.
Great. Jessica, Maddison, Hannah, thank you. That was really interesting. One question I have before we get to the chat, and you know, coming from someone I've squeezed a lot of fish in my days, even though I'm a water quality person. And I sometimes wonder, like 100 years from now, will our ancestors think we were so barbaric in how we handle, you know, other species, you know, in order to sample them. And so, you know, my question gets back, you mentioned the potential tag recapture. You know, would a beaver be a potential like pit tag type species where that could be a you know, an effective way of tagging and, you know, getting a better understanding of populations and movement? And I'm looking towards some pit tag experts in the room also, if you guys aren't aware of that. Has it ever been brought forward for beavers to pit tag beavers? Okay, it's been discussed. Just kind of wondering about that. And then I guess another bigger question in terms of dam removal, is there any relocation of beavers planned post dam removal? Has that been in the works?
Yeah, so I think for the first part of your question, we are really in the process still of trying to figure out if we can do different traps and see what methods are out there for more non-invasive capture. Because if we were able to look at like hair snares or something and do eDNA on that might be helpful. But that's kind of something that we're reviewing the literature about now and kind of seeing how that might play a role in the future. And I think potentially Jessica might be able to talk more about that second question about relocation.
Yeah, I don't know too much about it as like far as like for the state goes or anything like that. It's something that we've thought about, but our program here at Karuk DNR, but it's also something like we feel like we would need more information on like the current distribution of beaver. So like that's what this project is kind of like setting that baseline for.
Cool. Yeah, that makes sense.
So we were more informed. Yeah.
Great. There is a comment in here from Clint Alexander with a new eDNA paper that I hope is informative if folks want to take a look at that. It's not really a question, so I won't repeat it. But there is one question from Sarah Bjork from ODFW. Can small drone footage be used in lieu of walking surveys?
Yeah, that's a great question. Definitely, that could be a great future avenue to go down. Kind of like we mentioned, since this was such a piloting process, we wanted to get a better idea of where to prioritize those kinds of efforts, just because it still does take the capacity and being able to get those drones. So we want to know kind of which creeks or which areas to do that kind of those extra efforts. So that's definitely a direction that we could go in the future. And hopefully this research will kind of inform where we can even put those drones or even future game cameras, which is something that we did do there. Jessica and the wildlife crew put out a game camera where we did see those signs. So that was just a really exciting way to affirm what we were seeing, and potentially drone footage could be used in the future as well to do a very similar thing.
Hi, Betsy Stapleton with the Scott River Watershed Council. And I'm just speaking that the California Department of Fish and Wildlife has recently established a beaver unit to look at some of these issues and potential relocation and kind of supporting beaver as part of their nature-based solution initiative. And so I think we're going to be seeing a lot more effort from CDFW in answering some of these questions of understanding population and management.
Great. All right. If you don't mind stopping your screen sharing now, that would be helpful. All right, and I don't see any other hands raised. So thank you once again, Maddison, Hannah, and Jessica. All right, appreciate it.